let's move on. Okay. Next speaker is Anton de Kroon from Utrecht University. He's an expert in protein phospholipid interactions. And the title of his presentation is The Preferred Acyl Chain Donor of the Yeast Tafazzin. So he'll give us some information on phosphatidylserine involvement in the tafazzin reaction.
Okay, thank you, Grant. First of all, I would like to thank the organizers for the opportunity to present our work here. It's really been stimulating to interact with experts in Barth syndrome, both patients and researchers, especially coming from a lipid biochemistry lab. Okay, my group is interested in how organisms regulate their membrane lipid composition. In particular, we're interested in the interplay between phospholipid class and acyl chain composition. And we like to find out how organisms keep this composition such that important physical parameters stay within range. So we do this work in yeast for all the obvious reasons, including the limited repertoire of acyl chains in yeast and its unsurpassed tolerance to variation in lipid composition. Okay, this slide serves just to fresh up your memory. So yeast has a overall phospholipid class composition that's similar to mammalian cells. Its fatty acid composition is much simpler. There's basically 3 major acyl chains found. So the question I want to address today concerns the acyl chain donor of TAS1. So yesterday we heard all about synthesis of cardiolipin in yeast. It's localized to the mitochondrial inner membrane. Also, the cardiolipin-specific phospholipase turns out to be localized to the inner membrane, as we learned yesterday. And remarkably, the transacylase TAS1 seems to have a dual localization facing the intermembrane space and localized both in the inner membrane and the outer membrane. So now our question is, which is the substrate of TAS1 in yeast cells? So which is this phospholipid? So who are the suspects? Looking at the overall mitochondrial phospholipid composition, there's a little bit of PS, there's some 10% of PI, about 40% each of PE and PC. And PC really is the phospholipid that brought us into raised our interest in TAS, because we've been investigating remodeling of phosphatidylcholine in yeast, and I should give you a little bit of background about that first. So PC in yeast, as in mammalian cells, can be synthesized by 2 pathways. There's the methylation of PE, and there's the CDP choline pathway. And in the past, we showed that each pathway synthesizes PC with its own specific distribution of acyl chains. And if you add up these distributions, you don't end up with the steady-state PC species contribution, which is depicted in red here. Also, if you so that implies that you need additional processes, and this turns out that there's remodeling going on of PC. Also, if you knock out one or the other pathway, you still end up with the regular steady-state PC species distribution, and that's due to remodeling. So how do we measure remodeling? We do that conveniently in a strain that lacks the CDP choline pathway, so only PC from PE methylation is synthesized. Then we pulse the cells for 10 minutes with deuterated methionine, and in those 10 minutes, a subpopulation of PC is produced, which has this heavier head group, and using that heavier head group, you can selectively visualize the newly synthesized population of PC molecules by parent ion scanning mass spectrometry. So this approach has been coined dynamic lipidomics. So this slide shows a typical result. Relative abundance of the 4 major PC species. The nomenclature has been introduced yesterday by Fred. In the leftmost column, you see the distribution after 10-minute pulse. And after the 10-minute pulse, we exchange the deuterated methionine for regular protonated methionine, and we follow the fate of the deuterated population after different times of chase. And typically what we observe is a rise in 32.1 at the expense of a decrease in 34.2, which would correspond to the incorporation of a 16.0 fatty acid instead of an 18.1 fatty acid. Okay, as a biochemist, you like to interfere with the process, so we went on to search for genes that were involved in this process, which disrupted the process. Well, I can already give away that TES was not among these genes, but there were several others, and one stood out, and that was the SET1 gene, also known as GAT2. If we delete that gene, then we virtually abrogate PC remodeling, as you will appreciate by comparing these two graphs. So what's SCT1 doing? SCT1 encodes a glycerol 3-phosphate acyltransferase that's responsible for the first biosynthetic step in glycerolipid synthesis. So it attaches the acyl chain to the SN1 position of the glycerol 3-phosphate, yielding lyso-PA. Okay, so our first impression was maybe this SCT1 can also attach acyl chains to lyso-PC or to glycerophosphocholene. This turned out to be wrong. It turns out that by taking away SCT1, you modify the overall fatty acid composition of the yeast cells. Specifically, by deleting SCT1, you reduce the content of C16 by about 50%, whereas if you delete the homologous enzyme that catalyzes the same step, if you delete that, there's no large effect on overall acyl chain composition. Conversely, by overexpressing SCT1, we dramatically increase the C16 content at the expense of the unsaturated acyl chains C16-1 and C18-1, and the Western blot shows the level of overexpression. So we concluded that the defect in PC remodeling was due to the C16 content becoming limited for the remodeling. Okay, how does it work? So we propose that C16-CoA, which is produced by the fatty acid synthase in the cells, well, usually most of it will be desaturated by the single desaturation yeast encoded by the OLE1 gene, yielding C16-1-CoA. But as you overexpress SCT1, more of this C16-CoA is shuttled into lipids, and it's no longer accessible to the desaturase, and in that way you change the balance between saturated and unsaturated acyl chains in yeast. So there's competition between OLE1 and SCT1 for substrate. Okay, now upon overexpression of SCT1, the cells still grow happily, and this provided us with the opportunity to really characterize the cells, and we found that this increased C16 content is found back in all major membrane lipids, as you will see in this slide. So in PC, you see an increase in 32.1, 32.0 appears, PI 32.0 appears, so you actually see disaturated species appear throughout the lipid classes, and this also applies to cardiolipin. So here the empty vector control with the regular cardiolipin spectrum. If you overexpress SCT1, then there's a shift to the left side, so C16 gets enriched in cardiolipin as compared to the wild-type control. And you can also see that the degree of saturation increases by looking at the inserts, where the species with 3 and 2 double bonds now increase with respect to the species having 4 double bonds. Also seems that under conditions of SCT1 overexpression that the PG level goes up. Okay, so with this SCT1 overexpression, we have a new tool to manipulate yeast acyl chain composition, and we tested this also in remodeling experiments. So here again, we performed a pulse-Yates experiment using deuterated methionine, and we monitored the fate of newly synthesized PC. And well, there's quite extensive remodeling, particularly if you compare it to the wild-type situation where the effects were relatively modest. Here they are pretty large. So this provides us with a more sensitive tool to screen for the genes that are involved in the process. And for instance, when we deleted a phospholipase B encoded by the PLB1 gene, then we observed that steady state distribution of PC was not really affected, but the rate at which the newly synthesized PC evolved to the steady state was strongly delayed. And it's more obvious when we superimpose the arrows from the wild-type situation. So in principle, the approach works. And we then tested this system to look for the influence of TES1 on PC remodeling. And as is shown here, there's really no effect of TES1, the presence of TES1, on the rate of remodeling. Looking at the steady state of PC, there's maybe minor effects of the absence of TES1. However, if you look at the profile of the newly synthesized PC, that does appear to be an effect, because here the 34.2 level is much higher than in the control situation. So that suggests that the precursor of PC, PE, might be affected by deleting TES1. And that's shown on the next slide, where we took this SCT1 overexpression system and looked at the effect of deleting TES1 on the species profile of the major lipids. So PC shows minor changes, PI shows hardly any changes, and the changes are basically found in PE and PS. So in PE we see this enrichment of 34.2 in the absence of TES1. This is recapitulated in PS, where we also see a strong decrease in the disaturated 32.0 and 34.0 upon taking out TES1. So by deleting TES1, the level of unsaturation of PS and PE is increased. Okay, let's have a look at PS in yeast aminophospholipid metabolism. So PS is synthesized in the ER, and particularly in the mitochondria-associated membranes, where this enzyme is enriched. Then large parts, most of the PS is transferred into the mitochondria to be decarboxylated to PE. Part of the PE will stay in the mitochondria, part will go back to the ER, where it could be methylated to PC. Smaller part of PS migrates to the Golgi vacuole, where another PS decarboxylase is present. Okay, to get further insight into how TES affects PS and PE, we decided to start deuterium labeling studies using deuterated serine and follow the fate of newly synthesized PS. So upon pulse labeling for 20 minutes of wild-type cells, we got the following results for deuterated PS and deuterated PE. So we use specific scans that selectively visualize the deuterated populations of PS and PE. So in PS we see 32.2, 34.2 enriched as compared to the monounsaturated species, and this enrichment is stronger in PE, which is also synthesized in this 20-minute period. So the PS we are looking at here is also the leftover, which has not been decarboxylated. So it has been quantified in the next slide. So in wild type, we have a specific distribution of the newly synthesized PS, of the newly synthesized PE. And if you compare the PE to the PS, then you'll find that in PE, 2,2 is enriched as compared to the PS situation, greater than 34,2, whereas the monounsaturated lipids are definitely not enriched in PE, so apparently they are not preferred substrates for PS decarboxylation. In the test condition, it's more or less similar, but note again that there appears to be an enrichment in unsaturation in the newly synthesized PS population. So this result reminded me of an older paper from 10 years ago by the Finnish group from Pentti Sommerharju. He did similar experiments using baby hamster kidney cells, which he labeled with deuterated serine for an hour, and he monitored the conversion of PS to PE. And in this graph, he plotted all the molecular species going from the most hydrophilic species to the more hydrophobic species. And the more desaturated, the more hydrophilic, the shorter chains, the more hydrophilic. And as you decrease desaturation and increase lengths, the lipids become more hydrophobic. And he found that the more hydrophilic species are more easily converted into PE. In a control experiment, he took detergent solubilized PS decarboxylase and monitored the disappearance of PS during the decarboxylation reaction, and the PS, of course, decreased, but the species distribution in the decreasing population of PS molecules did not change. So he concluded that when PS molecules are more hydrophilic, they are more easily translocated into the mitochondria to be decarboxylated. Okay, back to our system. Conditions of SCT1 overexpression, so increased saturation. Comparing PS to PE, again, you see that PE is enriched for the more hydrophilic species, 33,2, 34,2 as compared to PS, and the disaturated PS species are not found back or barely found back in PE. Now, by deleting TES1, you reduce the relative content of saturated acyl chains in newly synthesized PS. So the 32,0 goes down dramatically, as does the 34,0, and 34,2 and 32,2 go up strongly. So how to explain this result? Well, the most straightforward explanation would be that TES is a consumer of PS. If TES were a consumer of PS, it would do so in the mitochondria, and it would preferentially take out the more hydrophilic, the di-unsaturated species, and leave the saturated ones intact. Unfortunately, we do not yet have internal standards of deuterated PS, so we cannot really quantify the populations. But fortunately, there's Miriam Greenberg's lab, and in the past she performed radio-labeled phosphate pulse labeling of wild type and TES deletion strain, the TES deletion strain. And remarkably, she found that after a 15-minute pulse, that the amount of label found back in PS is about twice as high in a TES1 mutant as compared to a wild type, which would be fully consistent with TES1 consuming the newly synthesized PS coming into the mitochondria. Okay, one final piece of evidence in favor of TES1 consuming PS. This is maybe a bit complicated. In this graph, we compare the steady state species distribution of PS to the pre-existing species distribution of PS in labeling experiments. So in labeling experiments, you introduce new PSs which are all deuterated, and then you can also look at the old protonated PS molecules present. And in our wild type background, we see that the steady state distribution is pretty much similar to the pre-existing PS population, as you would expect. Now, in the TES deletion condition, we see that there's quite a difference between the steady state and the pre-existing population. So, for instance, 34.0 is much lower in the steady state as in the pre-existing population. The same goes for 32.0, whereas 34.2 is higher. So this would indicate that that when TES is not there and when PS is not consumed, you have a larger population of PS, and it takes longer for PSD1 to completely process that. And therefore, the maturation of the PS population is somehow delayed, which further supports our proposal, which is then schematically depicted here. So we think that PS serves as a substrate for TES1 in re-acylating monolysocardiolipin. Then lyso-PS would be the side product. Lyso-PS could be either degraded by phospholipase or it could be re-acylated by an acyl transferase. And to conclude, so we showed that by overexpressing SCT1 and by manipulating the yeast acyl chain composition, we create a useful tool for dynamic lipidomic studies in yeast, which sort of increases the resolution of the effects you can observe. Furthermore, we show that it's likely that, like in mammalian cells, the decarboxylation of PS to PE decreases with increasing hydrophobicity of the PS substrate, which is consistent with more hydrophobic species being are less easily translocated into mitochondria. And the effects of TES deletion are consistent with TES1 consuming PS. Of course, data do not exclude other acyl chain donors. In fact, recently we did an experiment with a CO1 deletion mutant, which lacks the PS synthase. And in this mutant, we did not see any accumulation of monolysocardiolipin, so indicating that other donors are possible. So what are implications? Well, the fact that PS comes in the mitochondrial outer membrane sort of gives a rationale for the dual localization of TES1. By positioning TES1 close to the site where its donor comes in, it can readily compete with PSD and get the acyl chains it wants. Furthermore, it may give some selectivity to TES1. In yeast, there's no acyl chain specificity known for this enzyme. And by using the incoming PS, which is enriched in hydrophilic, this is unsaturated acyl chains, you sort of enrich the transacylation reaction, and you have a tool to increase the desaturation, the degree of desaturation of cardiolipin. Okay, then I would like to end by thanking the people who did most of the work. So Cedric de Smet, during his PhD research, did a lot of work here. He's currently, Tom Ewing is stepping in his footsteps, and he's helped by Ruud Cox and Tine Michels. We collaborate with Jos Brouwers from the lipidomics facility in the veterinary school, and with Femke Stedt and Fred Vaas at the University of Amsterdam. And funding is provided by the Barth Foundation and by NWO. And I thank you for your attention.
Green. Questions for two? Steve?
You mentioned in the CO1 mutant that there was no accumulation of monolysocardiolipin. Did you look and see if there were changes in the composition of cardiolipin?
Good question. There are changes in the composition of cardiolipin, and remarkably, this is all preliminary data. We only did the experiments once, but it seems that the C18 content goes up as compared to wild type. So an unexpected result, but we will look into that further.
This is really interesting. I was wondering if you thought about the relationship between PS flipping in apoptosis. Would that eliminate the PS inside the cell or inside the ER perhaps, so that there would be less available to TAS? Or would there be any relationship there?
Hard to tell. Apoptosis in yeast is controversial.
Oh, okay.
It happens in bacteria now.
Okay. Still. I don't know. I really don't know. Can only speculate.
Do you think that this localized generation of LPS that you'd see probably at the surface of the mitochondria might cause some, fluid dynamic effects, causing interaction with ER and mitochondria maybe to promote remodeling?
I doubt it. I think the lysolipids in general will be very short-lived because they are deleterious to membrane integrity. So I think the system probably has found ways to get rid of it very fast.
Okay. Any other questions? Okay, if not.